Hi, my name is Peter Kelmenson with Technical Information Services at the Jackson Laboratory. Thank you for viewing this presentation on Beyond the Knockout, using Cre-lox technology to model cancer, neurodegenerative diseases, and more.
Cre-lox technology is widely used to create traditional tissue-specific and inducible gene knockouts, but the technology is also a powerful tool for creating very sophisticated mouse models related to human diseases, such as cancer and neurodegenerative disorders. In this presentation, we will focus on some of the examples of how Cre-lox has been used to model human disease. We'll also demonstrate the power of Cre-lox as a reporter system to label specific cell populations. This is just a basic overview of what we'll cover today. I'll first talk about the basic Cre-lox mechanism, then I'll briefly discuss a couple Cre-lox breeding schemes comparing the knockout generation versus the gene activation breeding scheme. Then I'll talk about how Cre-lox can be used in human disease models of cancer and neurodegenerative diseases. And then I'll talk about the use of Cre-reporters for tissue-specific reporters and lineage tracing, and then some of the Cre-lox tools that they exist for cell biology type work. So the Cre-lox system was discovered as a natural part of the P1 bacteriophage viral life cycle, in which the viral DNA is injected into the bacteria, and then it gets circularized using the Cre-lox system, and that allows it for replication and development of new viral particles. It's a relatively simple two-component system, the first being the Cre-recombinase, which is a site-specific enzyme that's going to catalyze recombination between two LOXP sites, and symbolized by that sort of scissors designation we have in this presentation. The LOXP site is a 34 base pair DNA sequence. It's got an 8 base pair core sequence flanked by 2 inverted repeats, and it's the location and the orientation of those LOXP sites that determines the recombination result. And recombination can be a deletion, which is the most commonly used use of the Cre-lox, but also can be used to generate inversions and translocations as well. And the Cre-lox system allows for abundant possibilities for genome manipulation, as you will see throughout this presentation. So the basic mechanism for deletion is a gene or part of a gene is flanked by LOXP sites, often called floxed for flanked by LOXP. And then the Cre-recombinase is going to recognize those two LOXP sites and then recombine them so that you're left with the DNA to be deleted left as a circularized piece of DNA and a single LOXP site left on the chromosome, thereby generating a knockout or a null allele. So to generate tissue-specific knockouts, you typically start with breeding a homozygous LOXP flanked mouse with your gene of interest and cross to some sort of tissue-specific Cre. In this case, it's an albumin Cre, which has liver-specific expression of Cre. And from that cross, you'll generate mice that are heterozygous for the floxed allele and canned heterozygous for the Cre. So in those mice in the liver, the Cre is expressed and can delete the one copy of the floxed allele, but you still have another wild type allele left, so you haven't actually generated a knockout in the liver yet. It does require a second cross. So you take that same mouse that's heterozygous for the LOXP flanked allele carrying the Cre, and then you'll mate that mouse back to your homozygous LOXP flanked mice, and from that cross you will generate mice that are homozygous floxed and carry the Cre. And then in those mice, then the Cre is expressed in the liver, can delete both copies of your gene, thereby creating your liver-specific or your tissue-specific knockout. And what you expect from that cross is to generate mice that are 25% of the mice you generate will be homozygous for your floxed allele and carry the Cre. Now, I'd just like to compare that in a moment to what are called the lock-stop-lox alleles, or LSL. This is sort of the basis for a lot of Cre-reporter alleles, as well as many disease models using the Cre system. And so what one does is generates a promoter of interest, whether it's a tissue-specific promoter or, say, a widespread promoter, expressing some sort of gene of interest. But expression of that is blocked by a LOXP flank stop cassette that is in between the promoter and the gene of interest. So as that allele itself cannot express your gene of interest. However, whenever Cre is expressed, it will delete the stop cassette, thereby turning on expression of the gene. So therefore you can regulate turning on that gene using Cre, whether it's a tissue-specific Cre or an inducible Cre, or however that can happen. And so that's often used for Cre-reporters, as you can see in the top example, where you have, say, a LAC-Z expression blocked by that lock-stop-lox allele, cross to a Cre, then you get LAC-Z expression. But you can also use it to induce expression of any sort of transgene you want, as in the bottom example, say, is a disease model for cancer, where you have your promoter of interest expressing then a KRAS G12D mutant, which is an oncogenic form of KRAS. And then you can turn that on in a tissue-specific manner, generating cancer models or tumor models in whatever tissue you want. And just an example of how this sort of breeding can go, you have your Cre strain of interest, in this case a PDX1 Cre, which is expressed in the pancreas, and you cross that to then that KRAS G12D mice, and then in one generation you'll generate double transgenic mice where now the KRAS G12D is expressed in the pancreas, developing pancreatic tumors in one generation. So when those two strains are crossed together, you will generate in those double transgenic mice, they develop pancreatic interepithelial neoplasias, or PINs. Those are the precursors to an invasive pancreatic cancer. 100% of the mice will develop those, and some of those lesions will progress spontaneously to invasive and metastatic adenocarcinomas. And here you can just see some pictures of tumors, those neoplasms from those mice, as well as the development of the tumors at certain ages in time. So more recently, investigators at UCLA have actually improved on this model by including in the mice a LOXP-flanked PTEN allele. PTEN is a negative regulator of the PI3K AKT signaling axis, so it acts as a tumor suppressor. So in these mice that are heterozygous for the PTEN LOXP-flanked allele, heterozygous for that KRAS G12D, and have the PDX Cre, you can see that they develop pancreatic tumors in terms of they actually develop pancreatic ductal adenocarcinomas at a relatively good rate. And you can see here in that green line going down the graph their survival, and then you can also see the picture of the tumors in those mice as compared to in a human patient as well. And in fact, these KRAS G12D flox mice can be used to model early stage lung cancer alone. In fact, what you can do is you can introduce Cre nasally using adeno-Cre virus, and those mice will then develop lesions about 6 weeks post-Cre administration and develop into full adenocarcinomas by 16 weeks post-Cre. And you can see in those pictures where those tumors are generated in the lung, as well as a zoomed in photo of how those tumors look. So in addition to having different mouse strains that can be crossed together to create cancer models, the Jackson Laboratory has also imported some mouse strains that already carry multiple mutations in Cre transgenes together to work as cancer models, as is this example of mammary cancer, in which these mice carry a LOXP-flanked allele BRCA1. BRCA1 is a gene in humans that is very frequently found to be mutated in female patients that develop mammary tumors. And so in these mice, 64% of the mice that are homozygous for the BRCA1 floxed allele, heterozygous for the P53 knockout allele, and carry the Cre driven by the sheep beta-lactoglobulin transgene, so it's expressed in the mammary tissue. They will develop mammary tissues with characteristics of human basal-like breast tumors, which is a specific type of human breast cancers that typically strikes patients at younger ages and is associated with the worst prognosis for BRCA1 related tumors. So BRAF is another protein that's often found to be mutated in human cancers. BRAF is part of the RAS-activated RAF-MEK-ERK mitogen-activated protein kinase signaling pathway. When activated, mutant BRAF is detected in many human cancers like melanomas, colorectal cancers, thyroid, ovarian, and non-small cell lung cancers. The BRAF V600E is the most common mutation that's found. However, the progression to malignancy or tumors does require silencing of a tumor suppressor like PTEN as well. And this just shows you the sort of the pathway of where BRAF fits in. It's part of this pathway. It's RAF here in purple, this picture, and mutations that activate it then drive cell growth and proliferation. And then PTEN is a negative regulator of the PI3K AKT pathway. So when PTEN is inactivated, that removes that negative suppression and then leads to tumor growth. So here's a strain that the Jackson Laboratory has imported for melanoma model. And in these mice, they have a BRAF mutation with a LOXP-flanked allele. And so the allele they have is described here, where the normal gene or the normal exons are expressed, and you can see it goes from 14 and then jumps to a combined 15 exons, 15 through 18, all there. that would then give normal BRAF expression. But when Cre is expressed, it removes those exons 15 through 18, leaves one LOXP site, and then it has the mutant exon 15, which carries that V600E mutation, and then continues on to normal exon 16, 17, and 18 as well. So in these mice then that are het for the BRAF mutation, homozygous for a P10 LOXP flanked allele, and in this case they have a tamoxifen inducible Cre driven by tyrosinase, so that's expressed only in the skin. So then when you give those mice topical application of tamoxifen, turns on Cre expression, then converts to the BRAF V600E mutation and deletes P10. And in those mice, you'll get highly pigmented lesions that are detected 70 days post-treatment, as you can see in the picture D over to the right. And they develop to rapid progression to acute malignancy, and they don't survive very long post-treatment. And they will develop metastatic tumors to both the lymph nodes and the lungs. Now these mice will develop 25% or so or more will actually develop the melanomas without the tamoxifen by about 6 months of age, which indicates that the Cre itself is just a little bit leaky as well. So now I'd like to talk about the tetracycline inducible system and how that can work with the Cre-lox system as well to generate mouse models for studying human disease. So the TET system starts with a TET-O promoter, which has 7 TET operator sequences along with a CMV minimal promoter, and that then is driving expression of your gene of interest, but it will not be able to turn on your gene until the tetracycline activator protein is bound to that promoter. That's either the RTTA or the TTA version, which is a fusion of a TET repressor and a VP16. And it's the promoter then driving that tetracycline activator that determines the tissue specificity. So the RTTA, when doxycycline is administered, then allows binding of that protein to the TET operator, which will then activate your gene expression. That's known as the TET-on. TTA with doxycycline will prevent binding of the TET activator to the TET operator, and that will then turn off gene expression, and that's known as TET-off. And you can see this better diagrammed right here. So on the left side you have the TET-on system where you have your promoter of interest driving expression of RTTA. In the absence of doxycycline, that cannot bind to the TET operator and your gene is not turned on. On the right hand side is your TET-off system, same promoter driving transcription of TTA, that then can bind the TET operator and turn on transcription of your gene. When you give those mice doxycycline on the left, that will then allow the RTTA to bind the TET activator and then turn on transcription of your gene, so it turns it on. The doxycycline with the TTA will bind to it and prevent binding of the TTA to the TET activator, thereby turning off your gene expression, and that's why it's known as TET-off. And then you can use the tetracycline system to turn on Cre expression. You have your tissue-specific promoter driving RTTA, for example, say MMTV, where it's driving in a mammary tissue. It'll express RTTA, and then you have your TET-O Cre, and then in the presence of doxycycline, that will then bind and then turn on expression of Cre, which can then recognize LOXP sites in your gene of interest, recombining them, and then knocking out your gene of interest where it's expressed. Or it could be used to, say, delete a LOXP flank stop cassette and turn on your gene of interest. So you have multiple options using this system. But again, you're going to have 1, 2, 3 different alleles involved, so therefore the breeding is going to be a little bit more complicated. You can also use the Cre system in conjunction with the tetracycline inducible system to use Cre to actually turn on RTTA expression or TTA. So again, you have your tissue-specific Cre, and then you have a strain that, say, has a widespread promoter like GT Rosa, driving expression of RTTA, but expression is blocked by a LOXP flank stop cassette. So those mice don't express RTTA, and then of course you have your gene of interest driven by the TET-O promoter. And so when you combine all 3 of those alleles, Cre will be expressed in your tissue of interest, can then remove the LOXP flank stop cassette, and then you'll get RTTA expression, and then when you give those mice doxycycline, that will then turn on expression of your gene of interest. This is a system that's frequently used because there are a lot of strains out there that have tissue-specific expression of Cre, not as many that have tissue-specific expression of RTTA, so then you can use this sort of a strain here in combination with all of the tools that are available for Cre. So for example, you could use something like the PDX1 Cre, which is expressed in pancreatic tissues, cross to the RTTA strain, and then you say have your TET operon driving expression of a mutant form of KRAS, the G12D again, as was described in the reference down at the bottom. And then when Cre is expressed, it will recombine, express RTTA, and then you can use doxycycline then to determine when to turn on expression of that KRAS G12D, leading then to pancreatic tumors. And this is a system that you can use in cases where, say, the Cre is turned on earlier in pancreatic development, or the promoter driving Cre is turned on earlier, but maybe you want to turn on the cancer later. So you can have the PDX Cre delete the stop cassette, and they express RTTA, but the RTTA won't bind until you give the mice doxycycline, because once the cell is starting to produce RTTA, it's not going to go backwards. So you can use that where, say, the timing of where you're promoter driving Cre and when you want to turn on the oncogene to develop tumors may differ. So you can use this sort of system to very tightly regulate when you get the tumors to start. And again, you could also use this with TTA as well. Again, you have your tissue-specific Cre, and you have your TET operator driving activation of your gene. And here you have a similar situation where you have a widespread promoter like GT-ROSA driving TTA, blocked by a LOXP flank stop cassette. And then when you combine all three alleles into one mouse, you can then, the Cre will be expressed, it will recombine and start to express TTA, which in the absence of doxycycline will turn your gene on, and then you could say then use doxycycline to turn it off, depending on what you need, whether you may want to turn it off at certain times to see then what happens after that point. So here's a more specific example, again using Cre to turn on expression of a mutant form of SOD1, which leads to symptoms of ALS in humans. So here you would use that system. You turn on TTA using Cre with your whatever tissues you're interested in. In the absence of doxycycline, then the SOD1 G93A mutant protein is expressed, leading to whatever those phenotypes, and that then can be turned off or prevented expression of the SOD1 mutant protein by the administration of doxycycline. And this is sort of an important model conceptually because that administration of the doxycycline allows one to temporarily silence expression of that mutant protein and thereby give researchers a way to observe the importance of that mutant protein in the progression and persistence of that disease phenotype, because then you can monitor it at certain times and see what happens when it's active at certain periods of time. Another example here is a strain that expresses a different mutant form of SOD1, the G37R, and they have a LOXP flanked allele of that. So those transgenic mice will then develop ALS, neurodegenerative pathology. They, loss of motor neurons, motor weakness, paralysis, and end stage by 9 to 11 months of age. But if those mice are bred to either a motor neuron or a macrophage specific Cre, that will slow the disease progression. So here you can see by crossing those mice to different Cre strains, you can then slow the progression of that phenotype, which then allows for the ability for researchers to be able to sort of tease out what's going on in certain cell types that contributes to the ALS disease. So here you can see the data of crossing those mice to either a motor neuron or a microglia specific Cre, and both of them slows that disease progression. But what that does is allow investigators to tease out which certain cell types are involved in the ALS phenotype, and by this data shows that expression of the mutant form of SOD1 in macrophages contributes to the disease phenotype. So that creates a tool that which researchers can use to help better determine what is involved in that disease and how potentially what cell types or therapies might be used for helping to provide treatments. So I brought up previously Cre-reporter strains using the widespread promoter with a LOXP flank stop cassette driving expression of some sort of marker. The one I showed earlier was LAC-Z, but there could be any sort of marker molecule, a GFP, YFP, D-tomato, red fluorescent protein. Any of those markers can be used for Cre-reporters. And so the typical Cre-reporter breeding scheme is going to be very similar to some of the disease models I showed earlier, where you have your tissue-specific Cre, in this case a nestin Cre, which is expressed in the nervous system, crossed to your Cre-reporter, and then in wherever the Cre is expressed, you'll delete the LOXP flank stop cassette, turn on expression, in this case of LAC-Z. And here you can see the picture of those mice then showing a nervous system expression of LAC-Z, indicating that the Cre is confirmed to be expressed in those expected tissues. You can also see some variations where you have what they call reporter switching. So again, you have your widespread promoter driving expression, in this case of a D tomato, and the D tomato itself is flanked by LOX P sites, but the stop codon is following the D tomato. And then after the second LOX P site, you have a different fluorescent marker, in this case GFP. So these mice, as they come, would express D tomato in pretty much every single tissue. When they're crossed to Cre, the D tomato is deleted, and then those cells will then express GFP. So it allows you to look at not only which cells express Cre, but also whether the cells you're looking at actually express the reporter molecule altogether, so that you can distinguish between those issues. And here you're just going to see an example of that strain crossed to, in this case, a widespread tamoxifen inducible Cre, and then you can see the cells that are red then have no Cre expression. The cells that are green would have Cre expressed, and you can see some of the cell overlap where they both are expressed, and you can see that how you move to more green cells after 7 days post injection versus 3 days, and how the percentage of recombined cells increases over time. And there are some other variations that you can use this sort of system, not just for Cre reporting on Cre expression, but also to turn on or off transgenes. So again, you can have your promoter of interest driving GFP, and then that's followed by a stop cassette, and that's all flanked by LOX P sites, and then following that is your transgene of interest. So when you cross those mice to Cre, the GFP is deleted and you express your transgene of interest. And that way then you can use, for all of the GFP positive cells, those are going to be Cre negative, or the Cre isn't in those cells, and those cells won't express your transgene. All of the cells that are GFP negative then would have expression of your transgene. And again, there's other ways you can do that, a variation where you have, say, just a LOX P flank stop cassette preventing expression of your transgene that's fused to GFP, and then when Cre is expressed, you get a expression of your transgene GFP fusion protein, so you can use GFP to monitor where your transgene is expressed. Or sometimes you now see it more frequently, instead of a fusion, you'll have your transgene and the GFP or whatever reporter molecule you're interested in separated by an IRES sequence, so you get a bicistronic message. So instead of a fusion protein, you get an RNA that will express both proteins from that. So there you get expression again following Cre of both your transgene and your marker molecule. So this has led to one of the most visually spectacular and probably most inventive use of the Cre-lox system, which would be the brainbow mice, and in which these researchers did is they put multiple fluorescent proteins in the transgene construct separated by pairs of incompatible LOX P sites. So in these mice, what will happen is, following expression of Cre, you can get 3 different outcomes. You either get red fluorescent protein, yellow fluorescent protein, or cyan fluorescent protein. So when that happens, then you could get, in this case it's expressed in the brain, you get individual neural cells that express either red, yellow, or the cyan fluorescent protein, and you can tell them apart, as you can see in this picture. So you can trace all the neurons, all the axons as well of individual cells. However, like many transgenes, when these integrate, you often get multiple copies of the transgene inserting in tandem into the same locus. So if you see on the left side of this figure, for example, if you had 3 copies of the transgene, well, every transgene is going to have the same choice, red, yellow, blue, which means that you'll actually have 10 different possible combinations, much like the pixels in your TV give you different colors. It's a combination of those. So then with those 3 copies of the transgene, you'd have 10 color variations that you, with the right equipment, you could distinguish. And now when we look at some of the brainbow strains that are maintained at the Jackson Laboratory, the one in the middle has 8 copies of the transgene, now gets 8 choices of red, yellow, and blue, and up to 90 different color variations. And then the strain on the far right has more than 8 copies of the transgene and about 166 different color variations, which now gives a tool for researchers to look at individual neural cells and distinguish those cells and their axons and the pathway they go by from all of the neighboring cells purely based on their color alone, which is a really very powerful tool for looking at how neurons map and those patterns they lay down. And so that led also to the development of what they call the confetti mice, which is using the same transgene construct, but instead of being expressed by a neural specific promoter, is expressed by a widespread promoter, in this case GT Rosa. So therefore then when you cross that to any Cre strain in any tissues, then you can look at lineage tracing, because what's going to happen is you'll get different outcomes. You get yellow, red, or blue again. And in this case, when it was first generated, the investigators were looking at lineage tracing in the intestinal epithelium. And then what they want to know is if the stem cells in the intestinal crypts persist or do they compete with each other. And so what they were looking at is when those stem cells at the base of the crypt, when they divide, how can they tell whether they sort of go clonal or do they have just sort of general random population. So following across to a Cre strain that's expressed in the intestinal tract, after 1 week you can see there's a variety of colors, red, yellow, and blue. All of those cells are different colors. However, after 8 weeks you can see that now they're looking at more of a clonal population. So each of those little villi has either red, yellow, or blue instead of all the individual cells being multicolored. So it starts out sort of mixed but then moves to a more clonal population of cells, which is what they were investigating. And here you can see this just outlined. So as time goes on, those cells just became more clonal over time. The Jackson Laboratory has also imported a new set of Cre-reporter strains that were constructed by Dr. Hongqi Zheng and her colleagues at the Allen Institute for Brain Science, in which they incorporated different fluorescent reporters into the ubiquitously expressed GT-ROSA 26SOR locus, but also under the control of a strong exogenous promoter enhancer with the chicken beta-actin and the CMV intermediate early enhancer upstream of the LOXP flank stop cassette. And what those do is they give a brighter fluorescence when crossed to a Cre strain. As you can see, here's one example of one of those with D-tomato crossed to a CAMP kinase 2 to give neural expression. In addition to the fluorescent reporter mice, the Allen Institute for Brain Science has also developed a suite of Cre-responsive optogenetic transgenic mice. Optogenetic proteins are derived from light activatable bacterial opsins. They're channel proteins that, when expressed in neurons, can either activate or repress electrical signals depending on the types of ions they pass. And here you can see some of the strains that we have available listed here, the channel rhodopsin being the stimulatory ones, and the halorhodopsin and the archaeorhodopsin being the inhibitory ones. In the publication cited on this slide, the researchers generated several optogenetic reporter strains, including the strain you see up at the top there. These reporter strains were then crossed with a variety of different Cre strains, including the EMX1 Cre, which drives expression of Cre recombinase in the neocortex and the hippocampus. Following one cross of the optogenetic reporter with the EMX1 Cre, YFP expression was confirmed in the cortex and the hippocampus, and what you can see is that there was a similar voltage responses produced by either injection of current or with the light pulse, which is the purpose of the optogenetic models. And here's the same optogenetic model crossed to a different Cre strain, in this case a tamoxifen-inducible CAMP kinase 2A, and then you can see that when the light was off, the neurons would fire normally, but then when the green light was turned on, that would then inhibit neural firing. You can just see very few neurons then that just may not express Cre, were limited there. And then when the light came back on, the neurons would then fire again as normal. Some additional Cre-lox strains that can be used for research tools for cell biology are listed on this slide, including a strain that expresses a SUN1 or superfolder GFP MIC-tag fusion protein. SUN1 is a nuclear membrane protein, so this strain allows for purification of the nuclei via antibodies against GFP or the MIC epitope. The second strain expresses RPL10A, which is a ribosomal protein, which is a component of the 60S ribosomal subunit. So in these mice, also called FS-TRAP, activated GFP L10A labeled ribosomes are found in Cre-expressing tissues, and they can be immunoprecipitated to then pull out those active ribosomes. The third strain expresses a Dendra2, which is a green to red irreversibly photoswitchable monomeric fluorescent protein. Its fusion with a mitochondrial targeting signal of subunit 8 of cytochrome C oxidase generates a mitodendra2, which is then capable of mitochondrial specific fluorescence. And that's knocked in again to the GT-ROSA with a CAG promoter as well, and a LOXP flank stop that then conditionally that prevents expression until you cross it to a Cre strain. And the last strain allows for photolabeling of neurons in vivo. The Jackson Laboratory does maintain the world's largest collection of Cre-lox strains. We have currently, as of April 2014, more than 425 strains that express Cre, 170 plus which are inducible. We have more than 730 strains that carry a LOXP flank gene. More than 175 of those are with a flock stop or some sort of Cre-reporter. We also have more than 200 neuronal specific Cre strains, which you can find using that link right at the bottom of the page to search for. So you will find our Cre repository by coming to this page, either the link at the top of the page or from our website, you can either write on Find Jack's Mice, and that will pull up then this page, and you can click right there on that rectangle that says Cre-mice, which will pull up our Cre repository. There you'll find lists of the available Cre strains that we have, and then if you click that, you can then pull up sublists, whether you want inducible or LOXP flank sequence, etc. You'll also find the flip-frit strains that we also have as available. This presentation was made possible with your support. Jack's Mice and Services is dedicated to providing you with quality resources and flexible solutions and innovative technologies and unsurpassed expertise to support all aspects of your research. And realize that every dollar that you do spend at Jack's Mice and Services does support our shared research goals and sustains all these valuable mouse models and resources that are only available from the Jackson Laboratory. We do offer a host of services that might be helpful to you. Besides just having the specialty strains, we also do basic, custom, and complex breeding capabilities. So if you're having trouble breeding or you wish, we can breed Cre-lox mice together for you and either send you the breeding pairs or maintain cohorts for you here, as well as doing compound efficacy testing and genome scanning as well. And with that, again, thank you for attending this presentation. And if you do have any other questions, you can contact technical support at this link here, or you can contact your regional rep as well today if you want information from them, or schedule a visit if you'd like to get them in and talking to you directly.